Welcome to The Explainer. Today, we're diving deep, really deep, into the microscopic world. We're going to break down the fundamental unit of life in a way that's super clear and visual.
So let's just jump right in. I want you to imagine a single cell, not as some simple blob, but as this bustling, totally self-contained city. We're going to take a tour together, starting right at the city limits and working our way into its industrial heart.
All right, so our first stop on this tour is the city's outermost defense. We're talking about a massive, rigid structure that you'll find in plants, fungi, and prokaryotes. Welcome to the cell wall.
Okay, so don't think of this like a modern wire fence. It's much more like an ancient castle wall. It is tough, it's non-living, and its main job isn't really to pick and choose who gets in. Nah, its purpose is all about providing some serious mechanical strength and support.
So where does all this incredible strength come from? Well, it's all about the building materials. It's made mostly of these super tough fibers of cellulose. That's literally what gives plants the integrity to stand up straight. And it's even reinforced with other stuff like hemicellulose and pectin, kind of like rebar in concrete.
And what's really cool is how this wall is actually built. See, it's not just one slab. It's made in layers. It all starts with this shared middle lamella. Think of it like a layer of cement that glues all the neighboring city cells together. Then you get a more flexible primary wall for the young, growing cells. And finally, a thick, rigid secondary wall forms in mature cells, often beefed up with lignin for just maximum toughness.
But here's a really crucial detail. The wall gives it strength for sure, but it's also fully permeable. It's basically a fortress with the main gates stuck wide open. So, who's actually managing the traffic? Well, that critical job falls to the next layer we're about to meet, the real gatekeeper.
And that brings us right to the plasma membrane. This is the cell's super sophisticated border control. We're talking about a dynamic, almost intelligent barrier that carefully decides what gets in, what stays out, and even what gets shipped out of the city.
You know, the best way to really wrap your head around this membrane is through what's called the fluid mosaic model. And it was described with this one just beautiful phrase by the scientists Singer and Nicholson. It just paints the perfect picture in your mind. And this model is so important because it gets across a key idea. The membrane isn't some static, solid wall. Not at all. It's this flexible, constantly shifting mosaic of all these different molecules. It's fluid, almost like the surface of a thick soap bubble, which means all the little pieces can just drift and move around.
So what's this mosaic actually made of? Okay, so the foundation, the C itself, is this double layer of fats called the phospholipid bilayer. And then floating in this C, you have the protein icebergs. Some are integral proteins, meaning they go all the way through, acting like little tunnels or channels. Others are peripheral proteins, just sitting on the surface. You've got cholesterol tucked in there too, which helps keep everything at just the right level of fluidity. And those glycolipids and glycoproteins you see, think of them as unique ID tags on the outside so cells can recognize each other.
But the real genius of this whole thing is how it just automatically puts itself together. See, each of those little phospholipids has a water-loving head and a water-hating tail. So what happens? The heads naturally turn to face the watery environment, both inside and outside the cell, while all the tails try to get away from the water, tucking themselves into the middle. And boom, you have a stable, oily core.
And that oily core, that is the absolute key. It stops most things from just wandering through, which is what makes the membrane selectively permeable. So unlike the wide-open gates of the cell wall, this is a careful, selective gatekeeper that is actively controlling everything that crosses the border.
All right, we're in. We made it past border control. So now we find ourselves inside the main area of the cell, the cytoplasm. And as we look around, we can see this huge, interconnected system of factories and production lines, all humming along and working together.
What we're looking at is the endomembrane system. And it's not just one thing, right? It's a whole group of different organelles that all work together as one big coordinated unit. Basically, the manufacturing and shipping department for the entire city. So this system includes, the main factory floor, the packaging and shipping center, the recycling plant, and even the storage warehouses. For the rest of our tour today, we're going to focus on that very first stop, the main factory floor itself. And here it is. The endoplasmic reticulum, or just ER for short. I want you to picture this massive, sprawling network of folded membranes. It's like a maze of canals and sacs, and it's all directly connected to the nucleus, which is kind of like the city's central command.
This is where the real heavy lifting of production goes down.
The easiest way to think about this giant factory is that it has two different but connected departments. They actually look different, and more importantly, they specialize in making totally different products for the cell.
Okay, and here's the key difference you absolutely need to remember. You've got the rough ER, and it looks rough because its surface is literally covered with these little protein-making machines called ribosomes. That makes it the main assembly line for proteins. Then you have the smooth ER. No ribosomes, so it looks, well, smooth. Its job is totally different. It's more like a specialized chemical plant, making lipids and steroid hormones, and it's also where the cell detoxifies harmful stuff.
So, okay, our products, the proteins and lipids, have been made on the factory floor of the ER. But what happens next? I mean, how do they get sorted out, folded correctly, packaged up, and then shipped off to where they need to go? That could be another part of the cell, or even outside the city entirely. Well, that's where the next part of the endomembrane system comes into play. And that, my friends, is a story for...
All right, let's dive right in. Today, we are going on a tour, but not your typical one. We're shrinking down to go inside one of the most incredible, complex, and efficient factories in the universe, the living cell. So, let's get started. I mean, really think about that for a second. Everything from manufacturing to energy production to waste disposal, all happening inside a space so small you can't even see it. It's not just a chaotic soup of stuff, it's an incredibly organized, bustling operation.
So to really get a handle on how this whole thing works, we're going to get an exclusive look at 3 of the most vital departments. We'll check out how things get packaged and sent out, then we'll visit the cleanup and storage crews, and finally, we'll see the power plant that keeps the lights on for the whole operation. First up, let's head over to the cell's post office, its distribution center, its shipping hub. This place is called the Golgi apparatus, and trust me, its entire existence is about logistics. Yep, named after the guy who discovered it, Camillo Golgi. This organelle is basically a stack of flattened, membrane-bound sacks called cisterni. You can almost picture them like a stack of tiny, hollow pancakes. And their main job? To process, package, and ship out all the goods that the cell produces. And the Golgi is incredibly methodical. It's not a free-for-all. Materials like proteins and lipids have to come in through the proper entrance, a special receiving dock called the sys face. From there, they move along the assembly line of cistern, where they get modified and sorted. And once they're all ready to go, they get shipped out from the shipping bay, also known as the transface. This direction is super important and a key thing to remember. In at the sys face, out at the transface. So what's actually on the Golgi's job description? Well, it modifies the raw materials it gets. For example, it can add sugar chains to proteins, a process called glycosylation. Think of it like adding the final labels or gift wrap. Then it packages these finished products into little bubbles called vesicles and ships them off to exactly where they need to go. And super importantly, it also manufactures another key organelle we're about to meet, the lysosome. Okay, shipping is one thing, but any good factory needs a solid support system, right? You need a cleanup crew and a place to store all your supplies. Well, in the cell, those jobs belong to the lysosomes and the vacuoles. The division of labor here is pretty clear. Lysosomes are the cleanup crew. They are the cell's recycling center and garbage disposal all in one. Vacuoles, on the other hand, are the storage units. They're like the on-site warehouse, holding on to water, food, and other materials. Now that sounds dramatic, doesn't it? But it's a very famous nickname for lysosomes. Why? Because these little sacs are jam-packed with powerful digestive enzymes. They can break down old, worn-out cell parts, gobble up invading bacteria, and in extreme cases, if a cell is damaged beyond repair, they can actually digest the entire cell from the inside out. It's the cell's ultimate self-destruct button. Now, while animal cells have vacuoles, they really take center stage in plant cells. We're talking about one single vacuole that could become absolutely massive. And I'm not kidding about massive. Just look at this. A single central vacuole can take up to 90% of a plant cell's entire volume. It's surrounded by its own membrane, called the tonoplast, and it's filled with something called cell sap, basically water, sugars, and other stuff. By being so full, it pushes against the cell wall, providing turgidity. That's the internal pressure that helps keep plants standing up straight and not wilting. Okay, so all these departments, shipping, maintenance, you name it, they all need energy to run. Where does it all come from? Well, welcome to the power plant of the cell, the one, the only, the mighty mitochondria. This is probably the most famous nickname in all of biology, right? The powerhouse of the cell. And for good reason. Mitochondria are the site of aerobic respiration, which is just a fancy way of saying they take the food we eat and turn it into usable energy in the form of ATP. You can think of ATP as the universal currency of the cell. Without it, the whole factory just shuts down.
Let's take a quick look at the blueprints. Mitochondria have a double membrane. The outer one is smooth, kind of like the factory's outer wall. But the inner membrane, this is where the real action happens. It's folded into all these twists and turns called cristae. The whole point of these folds is to create a massive amount of surface area for making that precious ATP. And inside all of that is the matrix. Now here is what makes mitochondria so fascinating. The matrix has its own circular DNA and its own 70S ribosomes, totally separate from the main genetic code in the cell's nucleus.
This wild feature, having its own DNA and its own machinery to make proteins, is why we call the mitochondrion a semi-autonomous organelle. It's not fully independent, but it can make some of its own proteins whenever it needs to, without waiting for orders from the nucleus. It's like a department in the factory that can build and repair some of its own equipment.
So we visited all these individual departments, but the real magic, the true marvel, is seeing how they all work together.
Just think about the flow. The Golgi packages materials and also builds the lysosomes that handle cleanup. And the entire operation, every single step, is fueled by the ATP being churned out by the mitochondria. It's a beautifully coordinated, perfectly synchronized system.
But this brings us back to that really strange detail about the mitochondria, right? The fact that they have their own DNA? It kind of makes you wonder about the factory's origins.
I mean, think about it. You have this power plant operating inside a larger factory, but it has its own unique blueprints and its own machinery. Could it be that billions of years ago, mitochondria weren't part of the factory at all, that they were actually their own separate, free-living organisms that ended up in a partnership that would literally change the course of life on Earth? Well, all right, let's get right into it. Today we're taking a deep dive into the absolute heart of the cell. Think of it as the vault, the library, and the command center, all rolled into one. We are talking, of course, about the cell's nucleus.
You know, in any big operation, there has to be someone in charge, right? A CEO, a brain, something calling the shots. Well, a living cell is no different. So who is it? The answer is hiding inside this incredible structure that's part high-security fortress and part infinite library.
So here's the game plan for this explainer. We're going to start with the basics of the nucleus, then take a tour inside its fortress-like walls. After that, we'll see how all that genetic material gets organized, take a close-up look at a chromosome, and finally, we'll zoom back out to see how the nucleus works with other key players that really define a cell.
Okay, so first things first. Let's really nail down what this control center is and why it's so unbelievably important for, well, pretty much all life as we know it.
So, the nucleus. It was first spotted by Robert Brown way back in 1833, and it's usually the biggest, most obvious thing you see in a eukaryotic cell. It really has two main jobs. Job number one, protect the cell's genetic blueprint, its DNA, and job number two, control everything the cell does. We're talking growth, metabolism, making proteins, reproduction, everything. It is, without a doubt, the brain of the cell.
Now, let's do something cool. Let's actually go inside. You're about to see that the nucleus isn't just some simple bag. Nope, it's a fortress with some seriously impressive architecture.
Seriously, think of it like a fortress. It's got this double-layered wall called the nuclear envelope. And this wall has all these little gates, the nuclear pores, that are like security checkpoints, deciding what gets in and what gets out. Inside, you've got this gel-like fluid, the nucleoplasm. And right in the middle of it all, there's this dense, dark spot. That's the nucleolus. And you know, while the entire nucleus gets all the glory, if you had to pick a real unsung hero from inside those walls, it's gotta be the nucleolus. So what makes this little spot so important?
It's because this is where ribosomes, the cell's little protein factories, are actually built. The nucleolus is constantly churning out something called ribosomal RNA, or rRNA, which is the key ingredient for making ribosomes. And think about it. No nucleolus, no ribosomes. No ribosomes, no proteins. And without proteins, the cell just stops. So yeah, this little dark spot is a really, really big deal.
So we know the nucleus is protecting the DNA, but what does that DNA actually look like in there?
Because most of the time, it's not those neat little X shapes you always see in textbooks. Not even close.
See, in a cell that's just living its life, doing its job, the DNA is in a form called chromatin. And honestly, the best way to picture it is like a giant, messy ball of yarn. It's the DNA strand all wrapped around these proteins called histones. This messy, unwound state is actually super important because it means the cell's machinery can get in there and read the DNA instructions.
But just imagine trying to make it perfect copy of that tangled mess and then split it evenly between two new cells. It would be an absolute disaster. There's no way it would work. The cell needs a way to get organized before it divides. So, what's the plan?
The cell solution is nothing short of brilliant. It packs it all up. That loose, messy chromatin starts to condense and coil up into these super tight, organized, rod-like structures that we all recognize as chromosomes. This packaging only happens right before the cell divides, and it has one single purpose: to make the DNA easy to move so that each new cell gets a perfect, complete copy.
And this packaging job is just mind-blowing. Get this, a single human cell has about 2 meters of DNA. That's taller than most people, and it has to fit inside a nucleus that's only about 6 micrometers wide. How? Well, first the DNA wraps around those histone proteins, then those threads coil up, and then they supercoil, folding over and over again, until you get a chromosome that is thousands of times shorter than the original DNA strand. It's an incredible feat of engineering.
But this isn't just random packing. If we zoom in even closer on one of these fully packed chromosomes, we'll see it has a very specific structure, and every part of it is designed for the job of cell division.
Okay, so a chromosome that's ready to divide is actually made of 2 identical copies of itself. We call these sister chromatids. They're held together at this pinched-in spot called the centromere. And right on that centromere, you'll find these little protein discs called kinetochores. You can think of them as handles that the cell's machinery can grab onto to pull the sister chromatids apart.
And you know what's interesting? We actually classify chromosomes based on where that centromere is located. If it's smack in the middle, you get 2 equal arms, and we call that metacentric. If it's a little off-center, it's submetacentric. If it's way down near one end, it's acrocentric. And if it's right at the very tip, it's telocentric. This shape is actually super important for genetic analysis.
And hey, here's a great little trick from the source material to help you remember that first one. For metacentric, just think mes center mehun, which means I'm in the center. The name literally tells you where the centromere is. How cool is that?
Okay, time to zoom back out of it, because the nucleus and its chromosomes, they don't work alone. What they do, especially during cell division, is totally tied to other parts of the cell, organelles that can literally define what kind of cell it is.
Just take a look at some of the key differences between a typical animal cell and a plant cell. Now, that third line there is a huge one, especially for cell division. Centrosomes. Animal cells have them, but most plant cells don't. So what is a centrosome, and why is this such a big deal?
So, the centrosome is basically the main command center for organizing microtubules in animal cells. It hangs out near the nucleus and has these 2 little barrel-shaped things inside called centrioles. But its real moment to shine is during cell division. That's when it builds the spindle apparatus, this amazing web of fibers that latches onto the chromosomes and literally pulls them apart, ensuring each new daughter cell gets a complete set. And that right there is the key takeaway. The fact that animal cells have this centrosome gives them a very specific, centralized system for splitting up their chromosomes. The genetic plan inside the nucleus might be the same, but the machinery used to pull off that plan is fundamentally different. It's a perfect example of how cells have evolved different strategies to solve the same problem.
So we've seen how the nucleus is a vault, protecting the DNA blueprints. We've seen how it's a library, allowing access to those blueprints. We've even seen how it brilliantly packages them up for division. But that leaves us with one giant lingering question. What gives the order? What's the signal that triggers this whole incredible, complicated process to act?
Hey everyone, and welcome. Today, we're diving deep into the absolute fundamental unit of all life, the cell. It's the tiny engine that powers everything that lives and breathes, so let's get right into it.
Here's a simple way to think about it. You know how a huge skyscraper is really just made of thousands and thousands of individual bricks? Well, it's the exact same idea for living things. Every single organism is built from these microscopic units called cells. They really are the true building blocks of life.
So, here's our game plan for this explainer. We'll start at the very beginning with the first discovery,
Then we'll unpack the cell theory that grew from it. After that, we'll look at the two main designs for cells, zoom in on one of them, the prokaryotic cell, and wrap it all up with why the cell is the blueprint for everything. All right, let's kick things off with section 1, the first discovery. This is a pretty cool story about how something as simple as a piece of cork literally opened up an entire new world for science. Our story starts way back in 1665. A scientist named Robert Hooke was messing around with his new microscope and decided to look at a super thin slice of cork. What he saw were these tiny, empty, box-like shapes. They actually reminded him of the small rooms, or cells, that monks lived in, and believe it or not, that's how the name stuck. For more than 100 years, that was that. But then, in the 1830s, things got interesting. A German botanist, Matthias Schleiden, announced in 1838 that all plants were made of cells. Then, just 1 year later, his buddy Theodor Schwann said, hey, it's the same for animals too. And the final piece of this puzzle came in 1855 from Rudolf Virchow, who made the huge declaration that all new cells have to come from cells that already exist. So now we're moving on to our second section. You see, these weren't just a bunch of random observations. They were all coming together to form one of the most important, central ideas in all of biology, the cell theory. And together, these discoveries give us the 3 basic rules of cell theory. Rule number 1, every living thing is made of cells. Simple enough. Rule 2, the cell isn't just a building block, it's the basic functional unit. All the important stuff that keeps something alive happens inside cells. We can thank Schleiden and Schwann for those first 2. And then Rule 3, Virchow's brilliant insight. All cells come from other cells. This simple set of ideas unified biology in a way nothing had before. Now, whenever you have a theory this big and this bold, you have to ask yourself, is there anything out there that, doesn't play by the rules? And the answer is a big, resounding yes. The major exception you have to remember, especially for exams, is the virus. Viruses just aren't made of cells. They're basically just little packets of genetic code wrapped in a protein coat. They can't do anything on their own. They have to hijack a living cell to reproduce. They live in this wild gray area between living and non-living. This brings us perfectly to section 3, the 2 fundamental designs. So even though all true life is made of cells, it's not a one-size-fits-all situation. Life basically uses one of 2 major blueprints, the prokaryotic cell or the eukaryotic cell. And the difference between these two is night and day. On one hand, you have prokaryotic cells. Think of them as the minimalists. They're simpler, they're smaller, their DNA just kind of floats around in an area called the nucleoid, but it's not walled off. They also don't have all the little membrane-bound compartments, or organelles. Eukaryotic cells, on the other hand, are all about organization. They have a proper nucleus to protect their DNA and a whole bunch of specialized organelles that work like tiny organs, each with a specific job to do. So who belongs to which group? Well, the prokaryotic design is for bacteria and blue-green algae, the Monera kingdom. The more complex eukaryotic design, that's pretty much everything else. Protists, fungi, plants, and yeah, all animals, including us. Okay, time for section 4. Let's really zoom in and take a look inside a prokaryotic cell. Now, we called it the simpler blueprint, but don't let that fool you. This is an incredibly successful design that absolutely dominates our planet. So, the first thing you'll notice is its protection, the cell envelope, which has 3 layers. On the very outside, there's the glycocalyx, which is kind of sticky. Right under that is the cell wall, which is rigid and gives the cell its shape. And then the innermost layer is the plasma membrane, which acts like a bouncer, deciding exactly what gets in and what gets out. Now, a really cool feature of that plasma membrane is something called a mesosome. It's not a whole separate part. It's just an inward fold of the membrane itself. And why that matters, and this is a key detail to remember, is because these folds create a ton of extra surface area for important jobs like respiration and building the cell wall. So what's on the inside? Well, you've got the essentials. There's the nucleoid region, where the cell keeps its entire genetic blueprint on a single, circular piece of naked DNA. We also find ribosomes, which are the tiny factories that build all the proteins. And you might find some inclusions, which are basically little storage closets for extra materials. Let's pause on those ribosomes for just a second. In prokaryotes, they are a specific size called 70S. That S is just a unit that tells scientists about their size. Now, why does this one little number matter so much? Because our cells, eukaryotic cells, have bigger, 80S ribosomes. This tiny difference is a huge deal. It's the reason many antibiotics work. They're designed to attack only the 70S ribosomes, shutting down the bacteria without hurting our own cells. It's brilliant. On the outside, a prokaryote might have a few attachments. To move around, it could have a flagellum, which is this long, whip-like propeller made of 3 parts: the filament, the hook, and the basal body that works like a motor. Then you have pili, which are shorter tubes used to attach to things, and fimbriae, which are like tiny bristles that help the cells stick to surfaces. And that brings us to our final section, the blueprint of life. So let's zoom back out and think for a second about why this tiny little unit is at the heart of everything we know about biology. Rudolf Virchow said it best in Latin: omnis cellula e cellula, every cell from a pre-existing cell. It sounds simple, but it's a huge idea. It means every single cell in your body is part of an unbroken chain of life that stretches back billions of years, all the way to the very first cell. So the theory tells us exactly where all the cells we see today come from. But it also leaves us with this massive, mind-bending question. If every cell comes from another cell, where did the very first one come from? And that question pushes us right to the edge of what we know, reminding us that even in life's smallest building block, there are still huge mysteries left to explore. All right, welcome everyone. Today, we're going to go on a pretty incredible tour. We're not jetting off to a new country or some distant planet, but to a world that's just as complex and honestly, way more fascinating. The world inside a single cell. So let's dive in. I mean, just stop and think about that for a second. Inside every single living cell, there's this bustling metropolis. You've got power stations, factories, intricate highway systems, even a central government. And it's all working in perfect harmony, on a scale so tiny we can't even begin to see it without some serious tech. So, you ready? We're about to shrink down and get a real up-close look at the absolute marvels of biological engineering that, keep life itself running. Let's get started. Okay, our first stop on the tour is a facility you'll only find in plant cells. We're talking about plastids, a whole group of organelles that are responsible for some of the most critical and definitely most colorful jobs in the entire plant kingdom. So, what in the world are plastids? Well, think of them as the cell's specialists. They're these little membrane-bound packets that handle production and storage. They are literally the reason leaves are green, why carrots are orange, and how potatoes get packed full of starch. And they come in a few different flavors, each one with a totally unique job. Let's meet the first 2 types. So on one side, you have the leucoplasts. These are basically the cell's pantry. Super practical. They're colorless, and their whole job is just to store essential stuff, like starches and oils. But then, you've got the chromoplasts, and these are the cell's paint factory. They are packed with pigments that create those brilliant reds, oranges, and yellows we see in flowers and fruits. But, beyond the storage closets and the paint shops, there is one plastid that is the true superstar. It's the one that literally powers the entire plant world. It's an absolute masterpiece of natural engineering. And that, of course, is the chloroplast. This is the green gem of the cell, absolutely stuffed with chlorophyll. It's a microscopic solar power station, capturing sunlight and turning it into energy through photosynthesis. I mean, this one process is what feeds almost all life on Earth. No big deal, right? Now, harnessing the power of the sun, that's not a simple job. The chloroplast's structure has to be incredibly sophisticated to pull it off. So how is this little power plant actually put together? Let's take a closer look. Okay, so here's the basic blueprint. It's all wrapped in a protective double membrane. Inside that, you have the stroma, which is this fluid-filled space, kind of like the main factory floor. It's even got its own DNA and little ribosomes. Now, floating around in that fluid are the thylakoids, these tiny coin-shaped sacs where all the light-capturing chlorophyll is. And here is the genius part. To be as efficient as possible, these little coins are stacked up into towers called grana. It's just a brilliant design to soak up every last bit of sunlight. All right, tour's moving on. We're leaving the power plants, and now we're heading over to check out the city's infrastructure and its manufacturing hubs, the cytoskeleton and the ribosomes. So here we have 2 absolutely crucial components. First, the cytoskeleton. This is the cell's internal framework. It's like a combination of scaffolding and a highway system. This dynamic network of proteins gives the cell its shape, its strength, and it's also how things get moved around. And then you have the ribosomes, Oh man, these are the real workhorses. They're like tiny 3D printers that read genetic code and build every single protein the cell needs to function.
But this cytoskeleton isn't just one boring structure. It's this incredible dynamic network of different kinds of filaments all working together. You've got the big, hollow microtubules, which act like the main support beams. Then you have the thinner, more flexible microfilaments, which are more like ropes and pulleys that let the cell move and change shape. And finally, you have these super tough intermediate filaments that just add reinforcement, holding everything tight. Together, they create a structure that's both super strong and constantly changing.
And what about those protein factories, the ribosomes? Well, it turns out they come in two main models. You've got the ADS, which is the bigger standard version found all over complex cells like ours. But what's really fascinating is the smaller 70S model. You find it in simple bacteria, sure, but you also find it inside the cell's own power plants, the chloroplasts and mitochondria. It's a huge clue that points back to their very ancient origins.
All right, for our final stop, we're heading to the edge of the cell city to check out its engines. I mean, how does a cell actually get around? Well, it uses two amazing structures, cilia and flagella.
Okay, so both of these are these hair-like things that stick out from the cell and create motion. Cilia are the short, numerous ones. Imagine hundreds of tiny oars all rowing in perfect sync. Flagella, on the other hand, are much longer, and you usually only see one or two on a cell. They move in this whip-like way, kind of like a powerful propeller on a boat.
So you've got oars and you've got propellers. Two totally different ways to move, right? But here is the incredible part. If you could just slice one of each open and peek inside, you would find that their core engine is built from the exact same, unbelievably precise blueprint. And that entire blueprint can be summed up with one simple, almost poetic number, 9 plus 2. What on earth could that mean? It all comes down to the arrangement of microtubules inside their core structure, which is called the axoneme. This axoneme is the motor. It's the engine that drives all that movement. And its design is one of the most elegant, repeated patterns in all of biology. So here's how this 9 plus 2 blueprint works. In the motor part, the axoneme, you have a ring of 9 pairs of microtubules surrounding 2 single ones right in the center. That 9 plus 2 structure is flexible. It lets it bend and create that whipping motion. But of course, an engine has to be anchored down, right? That's the job of the basal body, and it uses a slightly different, more rigid structure, a 9 plus 0 arrangement with no microtubules in the center. That simple switch from 9 plus 2 to 9 plus 0 is the difference between a flexible motor and a solid foundation. It is just an incredibly elegant piece of engineering. And that's the end of our tour. We've seen solar power stations, we've seen protein factories, a dynamic city skeleton, and these incredibly sophisticated propulsion engines. And all of this machinery, all of these complex systems are working together perfectly inside a space too small for us to even see.
So I guess the big takeaway is this. A cell isn't just a bag of stuff. It's a city. It is a living, breathing, dynamic system where every single part is connected, every function is coordinated, and the whole thing works with an elegance that is just, honestly, it's breathtaking.
And that really leaves us with one last thought to take away. If this is the kind of complexity and engineering brilliance that's happening inside just one of your cells, what does that say about the trillions of them that are all working together